Have you ever wondered what a microbe really looks like? Not just a cartoon in a textbook, but its true intricate form. Every ridge, every spike, every detail magnified thousands of times. That's the magic of the Scanning Electron Microscope, or SEM. In today's video, you'll get a chance to learn about Scanning Electron Microscope, its parts, and the process behind this powerful tool, guided by Dr. P Palani, Director and Head of the Department at the Center for Advanced Study in Botany, University of Madras. Get ready to explore the unseen. microbiaX, microbe-inspired innovation.
I am extremely happy to share my knowledges with the operation of Scanning Electron Microscope on the occasion of International Microorganisms Day. There are different levels of Scanning Electron Microscope available for sample analysis. The one we have at the Center for Advanced Studies in Botany is Biological Scanning Electron Microscope. And the two other versions of scanning electron microscopes are field emission scanning electron microscope, and the next one will be the high resolution scanning electron microscopes. So let me first explain how this biological scanning electron microscope works and how to prepare the sample for this particular scanning electron microscope. First of all, this is actually the heart of the instrument. Inside of this we have a chamber, and the chamber is completely maintained under vacuum. And inside the chamber, the sample has to be placed and sample analysis has to be done. Before we could analyze the sample, let me explain the components of the biological scanning electron microscope. This is a small chamber in which electron generating bulb is available, and below that we have the electromagnetic lenses, and inside we have a stage where we can keep our samples for analysis. The chamber is completely maintained under vacuum. Now what is going to happen is, once the bulb is switched on, electrons will be generated, and the generated electrons will be in a dissipated form. And the generated electrons will go through the three layers of electromagnetic lenses which are available on the entire length of this part. And when the electrons are passed through the electromagnetic lenses, the electromagnetic lenses will focus the electrons, and a focused beam of electron will ultimately enter into the chamber. And we have already placed a sample in the chamber inside. When the sample is hit by electron beams, three different reactions will take place. And depending upon the nature of the sample, there will be reflection of electrons, and those electrons are called backscattered electrons. The electrons which are generated in the bulb are called primary electrons. Our sample is made up of so many elements, and each of the elements have electron in the outer orbits. So what is going to happen when the electron generated by the bulb hits the sample? The electron present in the sample will be dislodged, and those dislodged electrons will be the secondary electrons. And the third reaction will be generation of X-rays. So these are the three different reactions: backscattered electrons, production of secondary electrons, and the production of X-rays. These are the three different reactions which will take place inside the sample when the electron beam hits the sample. So there is detectors inside the chamber, and these detectors will detect how many backscattered electrons are coming from the sample, how many secondary electrons are coming from the sample, and how much of X-rays is produced. All these events will be recorded by the detectors, and these detectors will send the signal to the software, and the software will put all these signal as white and black dots, and ultimately we are going to have a clear magnified image of the sample. So one of the most important step in the electron microscopic analysis of sample is the preparation of sample. We have to make a discretion before we could analyze a sample, whether the sample is electron conducting or electron non-conducting. And as you are aware, most of the biological samples are electron non-conducting. If such is the case, all those non-conducting samples have to be converted into electron conducting sample prior to analysis of your sample into the electron microscope. We take a small aluminum stub. So this is an aluminum stub on which a carbon film will be pasted on the surface. On top of the carbon film, our sample to be analyzed has to be kept. So once the sample is placed on the carbon tape and the entire setup will be placed in a chamber. And see, this is a small unit. I already mentioned that the entire chamber has to be kept under vacuum. You can see a ring of a metal. Here we have the metal as gold, and it's a pure 24 karat gold. Instead of gold, the metal can be either platinum or silver or any metal. Any metal can be placed inside the chamber. Finally, the chamber will be closed, and now the machine will be switched on. Now the vacuum is generating. So we will have to wait till a sufficient amount of vacuum is generated within the chamber. So what is going to happen when the sufficient vacuum is generated? When a high amount of electricity is supplied to a metal, and the metal will vaporize under vacuum, and all those metal vapors will fall on the surface of the sample, and the entire surface of our sample will be covered with metal ions. So previously, the sample which was electron non-conducting, and after the sputter coating, the sample will be converted into electron conducting material. Now I am going to switch off the machine. So the sample will be coated with metal. We are going to take it out, and which will be analyzed in the electron microscope. These are all the processes which have to be done before sample analysis for scanning electron microscope. I also would like to tell you something about the preparation of sample. So once the sample is collected, it may be any sample. It may be plant sample, it may be bacterial sample, it may be fungal sample or animal sample, whatever it is. But we have to make sure that the sample is completely dry. There should be any kind of moisture in it. If moisture is available, then when we analyze the sample under electron microscope, your sample will be hit by the electron beam, and there will be the electron dissipation also will be there, and we may not get clear images of your sample. So therefore it is very, very important that our sample has to be completely free from any moisture. For example, for a small samples like fungal mycelia with spores on them, we can directly take those samples and place on the carbon tape, which will be placed on the aluminum stub, and directly we can go for coating the samples. After the coating, the sample can be analyzed under electron microscope. For complex organisms like plant tissue or animal tissue, the sample has to be fixed prior to the coating with the sputter coated and prior to analysis of the same sample under electron microscope. And how to do the fixation? The fixation is normally done with 2.5% of glutaraldehyde. When we put the sample in a solution of 2.5% glutaraldehyde, what the glutaraldehyde will do, the glutaraldehyde will crystallize all the molecules, macromolecules like protein, carbohydrate, and lipid molecules. And the original structure of the sample will be retained as such. So the fixation is normally done in 2.5% glutaraldehyde solution for about 24 hours. After fixation, The next step is going to be the dehydration. The dehydration is done by the treatment of your sample with a linear gradient of alcohol. The sample has to be treated with the low percentage of alcohol first, and then gradually the alcohol percentage has to be increased. And when we reach the final stage, the sample has to be treated with 100% alcohol. Alcohol will remove all the water molecules from the samples. After the treatment of your sample with 100% alcohol, there won't be any kind of moisture in it. So complete removal of water molecule will take place. Now, if it is electron non-conducting, then it has to be sputter coated with the sputter coating machine. As I informed you earlier, the sputtering process can be done with any metals. And this machine has gold as the metal, comes with the chamber. But instead of gold, platinum can also be used, silver can be used, and then other metals, similar metals can be used. Ultimately, when Butler's coating is completed, then the sample can be taken for SEM analysis, and we can get as many number of images as possible. Those images can be used for various purposes, including publications, writing, review article, and so many other purposes. For the sputtering process, the sample has to be placed on a carbon tape, and the carbon tape in turn has to be fixed on the aluminum stub. Why we have to place our sample on the surface of a carbon tape? So now the carbon tape is going to be placed on the metal stub. This is the carbon. We know that the carbon is a completely electron conducting material on which our sample has to be placed. After placing your sample on the carbon tape, the entire setup is going to be placed inside the chamber. Assume that this is our sample. Our sample is completely placed and is stuck on the carbon tape. Now it cannot fall. The carbon tape does two functions. One function is to finally fixes the sample on the surface of the carbon tape, and second function done by the same carbon tape is it is completely electron conducting material. The sample now has been placed on the carbon tape, and the next step will be coating the sample with carbon, gold, platinum, or any other metal. As I briefed earlier, and this machine has gold as a metal. The sample we placed on the aluminum stub is electron non-conducting sample. So therefore, it has to be converted into electron conducting material. So for that purpose, we are going to do the sputter coating with gold. So the sample is placed on the platform. There is a small platform inside the chamber, and now the top part of the chamber is going to be placed. We have a gold metal inside, and so this chamber will be closed, and the vacuum pump has to be switched on. The vacuum will generate, and it's going to take 2 to 3 minutes for a complete generation of vacuum inside the chamber. And the chamber now cannot be removed because the vacuum is developing inside the chamber. And so once it reaches the sufficient vacuum, sputtering switch has to be switched on, and the electricity will go to the metal ions. As I told you before, the vaporization of metal ions will take place when we apply very high amount of electricity to a metal under vacuum. The coating process will depend on the nature of the sample. Some sample will require more amount of coating. For example, like bacterial or fungal or viral samples, they are very, very minute samples, and they require the extended time of coating. And for samples like biological origin, like plants and animal samples, The sputtering process can be completed within 1 or 2 minutes. The amount of sputtering process depends on the nature of the cell. And once the sputtering process is over, coating process is over, the chamber has to be opened when the vacuum is released. So now I'm going to release the vacuum, and the vacuum now is gradually will be released, and the chamber is completely filled with the air now. Now the chamber can be removed. So this is the sputter coated sample, and this sample can be taken for SEM analysis. So this is about one of the ways by which we can prepare the sample. And once you come to know that the sample is non-electron conducting, then it has to be converted into electron conducting material. Once it is modified, the modified sample can be kept inside the electron microscope, and we can get as many images as possible with varying degrees of magnifications. So as far as the electron microscopic analysis of samples are concerned, the first and foremost step will be the preparation of a sample. These samples can be prepared in different ways, and it depends on what kind of preservation method, what kind of sample preparation method has to be used for what sample will be decided by the nature of the sample. For example, if you are going to analyze biological samples like plants or animals, those samples can be analyzed even without preparation of samples. The live samples can be used and analyzed without even any kind of preparation. But it is advisable to use the sample preparation step first. The most conveniently used preparation method is fixation is the first step, and the fixation of your sample is done with 2.5% chloride solution, and the duration of the treatment will vary from 1 hour to 24 hours, 48 hours, depending upon the nature of the sample. So what is going to happen during the fixation process? The macromolecules like proteins, carbohydrate and lipid molecules, they will be frozen. So the original structure of the sample will be maintained. The next step will be dehydration. The dehydration is normally done with a linear gradient of alcohol. We will start with 25% alcohol first, and which will be increased to 40%, 50%, 60%, 75%, 90%, finally 100% alcohol treatment. When the dehydration is done, the sample is expected to have no moisture in it. That is the quality of sample that we need for scanning electron microscopic analysis. Once the sample is dehydrated, then the dehydrated sample can be placed inside the sputter coating machine, and the sputtering process can be completed. Once the sputtering process is completed, the sample can be removed from the sputter coating machine, and which will be used for the electron microscopic analysis. I want to also tell you something about the preparation of sample, especially the plant sample. Most of the plant samples does not require the fixation, dehydration and sputter coating processes. Even without the fixation of a sample, the sample can be directly used for imaging analysis. Similarly, many of the animal samples, especially the insects, also do not require the sample preparation like fixing, dehydration and sputter coating processes, and those samples can be directly viewed under electron microscope. So these are all the advantages available with some of the biological samples. When we go for the analysis of bacterial fungal samples, bacteria require fixation, dehydration and sputter coating processes. When we come to the fungal samples also can be used as such. So what we normally do, we grow the fungal sample on a thin film of medium that is prepared on a cover glass. We allow the fungus to grow on it, and what we do, we take the entire cover glass with the fungus, grown fungus on the cover glass, and then immediately what we do, we freeze them, and once the freezing process is completed, then we will go for sputter coating with gold, and once the sputter coating is completed, the sample can be removed and can be analyzed in the electron microscope instrument. I just want to summarize the steps involved in the scanning electron microscopic analysis of any sample, not necessarily a biological sample, but it can be any type of sample. Even the polymer sample can also used. The first step will be the selection of the sample for imaging analysis, scanning electron microscopic analysis, and the second step has to be fixation. The sample fixation is the second step, where your sample will be fixed. When you do the fixation of a sample, all biological or metabolic activities of the sample inside the cell will be completely arrested, and you will get the complete originality of the sample. And the next step will be dehydration. And the dehydration is normally done with alcohol treatment. And the next step will be the sputter coating. The sputter coating is done with the machine, the sputter coating machine. Once the sample is completely sputter coated, we convert them into electron conducting material, and the electron conducting material can be analyzed under electron microscope.
microbiaX, microbe-inspired innovation.